All right, so this is the first lecture for Chapter 4, and you may have noticed Chapter 4 is a pretty large chapter, so we've broken it down into 3 lectures to hit each of the 3 main topics in the chapter. So in this first lecture here, we're going to talk about protein structure and protein function. And first we'll start out by talking about the building blocks of proteins, known as amino acids. And all amino acids have the same core structure. They have an alpha carbon here in the center, they have an amino group, they have a hydrogen, they have a carboxylic acid, and then they have an R group. And these R groups are what make each amino acid unique. Now this chart here shows the 20 common amino acids, and these are broken down into 3 groups based on the chemical properties of those R groups, those groups that make them unique. And you can see here, they all have this same core structure like you see here, but then they have these unique R groups, which are shown in red here. And these are what make each amino acid unique. So the first group, hydrophobic nonpolar amino acids, it makes sense. If they're nonpolar, they're more than likely going to be hydrophobic because water is polar. And so this collection of amino acids are hydrophobic nonpolar. And that makes sense. If you look at the structures here, they're mostly comprised of hydrocarbons. They're not charged. They're not able to form hydrogen bonds. So it makes sense that they're nonpolar and thus hydrophobic. And the second group here is uncharged hydrophilic polar amino acids. And now if they're polar, it makes sense that they'd be hydrophilic because water is polar. And the nature of their R groups make it where they're typically in the uncharged state. However, their side chains are able to form hydrogen bonds, which contributes to secondary structure of protein structure. And that makes sense. You see a lot of these alcohol groups, these amine groups, and these are what can form hydrogen bonds, which then contributes to the polarity of these R groups. And then lastly, the third group here are charged hydrophilic polar amino acids. So again, these are polar. It makes sense that they're hydrophilic. And the side chains carry a net charge at neutral pH, which if you remember from your chemistry days is 7. And the typical pH in the human body is actually 7.4, so pretty close to neutral. So it makes sense that in the human body, these amino acids are going to likely carry a net charge. And if you look at their R groups, they have amine groups here that can gain a hydrogen and form a positive charge. And then there's ones over here that have carboxylic acid groups that can actually lose a hydrogen and then turn into a negative charge. Then you have what's called the essential amino acids. And the reason they're called essential is because these are amino acids that need to be consumed in the diet because we cannot synthesize them in our bodies. So the only way we can get them is by eating them. And there's a nice mnemonic called Private Tim Hall that's used to help us remember this. And P stands for phenylalanine, V for valine, T for threonine, tryptophan, isoleucine, methionine, histidine. And then one thing you want to note here is that whoever made this up just put this A in there because they had to spell Hall. And A is just for the mnemonic. It doesn't represent an amino acid. And then you have leucine and lysine. Now this figure, you've seen it come up a lot in our lectures. And that's because it just outlines these core processes of biology. So first you have DNA replication. Then you have transcription, which produces the mRNA. The mRNA exits the nucleus, and that's where translation occurs, which is what we'll talk about over the next 2 lectures here. And translation involves essentially translating the mRNA sequence into an amino acid sequence, or a polypeptide chain that ends up turning into the primary sequence of a protein, which we'll talk about in great detail over the next few slides. All right, so protein structure is broken down into primary structure, which is here. This is the amino acid sequence that's actually produced directly from translation in the ribosome. And then you go from primary structure to secondary structures, which are these 2 here. These are beta sheets and then an alpha helix. So that's secondary. And then secondary can start folding and form tertiary structures. So you have multiple secondary structures here. And then in some proteins, you have multiple tertiary structures that combine as subunits to form one large protein, which is quaternary structure. So primary structure, like we said, this is produced during translation, and it's composed of a linear sequence of amino acids. So you have amino acids like this. They're each bound together like this, and they're bound by what's called a peptide bond. So as they're being synthesized, there's a peptide bond that's being created between each amino acid as it's added to the growing chain. And down here, we'll take a look closer at what the peptide bond is, because sometimes you can get tested on this. So you have 2 amino acids here. You have the carboxylic acid group here, which is joining with the amine group here. And as you can see here, you essentially remove a water, so an H2O. And then you form here the peptide bond, which is a covalent bond, as we say here. between the carbon in the carboxyl group and the nitrogen in the amine group. And actually, in the polypeptide chain, you have what's called the C terminus, which is here because you see this is where the carbon is, and then you have the N terminus. So these are just conventions of where it's kind of analogous to 5' end and 3' end in DNA or RNA. Same kind of thing. It's just to denote which end of the amino acid sequence you're at. And actually the convention is that the beginning of the amino acid sequence is actually considered the N terminus. And then the end of the amino acid sequence is defined by the C terminus. So here's a nice figure showing primary structure again. You can see these circles all represent amino acids. They're a linear sequence. Now the interactions that occur between these amino acids, like this and this and this, these are actually what determines the secondary and tertiary structures. Therefore, the primary structure or primary sequence actually determines the secondary and tertiary structures. And then as a result of determining those secondary and tertiary structures, it often determines the overall function of the protein because the protein's function is heavily dependent on the structure. Now, when you get to secondary structure, there's regions within the polypeptide chain that form hydrogen bonds, and they create one of the two following structures, alpha helix or a beta pleated sheet. So an alpha helix is a spiral-shaped structure, and it has the side chains of the polypeptide actually directed outwards. So the side chains would be the R groups. So you have R groups pointing out like this as you go up like this. And the hydrogen bonds that hold these together are, like so, between amino acids to help form this spiral-like structure. One thing to note is that the amino acid proline, remember it has that bulky ring structure as its R group. So there's no free hydrogen available to form hydrogen bonds. So if there's a proline in the amino acid sequence, it's going to disrupt alpha helix formation. So that's an important fact to know. Beta sheets or beta pleated sheets, these are formed by beta strands, which are a segment of polypeptide that are about 3 to 10 amino acids long. So here's one here, here's one here. And then they're connected laterally via hydrogen bonds, which are shown by these dotted lines here. Protein motifs are specialized secondary structures, or as we say here, super secondary structures. And they're composed of more than one secondary structure that form a three-dimensional shape. And that shape that's formed by the combining of more than one secondary structure is what correlates to the protein function. Now, these secondary structures that combine, they can be all the same type, so they can all be alpha helices, they can all be beta sheets, or they can be a combination of an alpha helix and a beta sheet. Two common examples that we'll go over in the next couple slides are zinc fingers and leucine zippers. So zinc fingers, these are a common example, and they're formed by a zinc ion, which we show here. And it's bound by two histidine, which are shown here. So this is a histidine. This is a histidine. Remember, it has that ring-like structure. And then two cysteine amino acids, so here and here. And so they essentially coordinate this zinc ion. So that's how that forms the structure. And these are frequently a component of receptors that have DNA binding domains, such as steroid hormone receptors. So if you have a steroid hormone, remember, it can have a region that binds the steroid itself. So the steroid comes in, we'll say this is a steroid receptor. The steroid comes in and binds would be the ligand binding domain. So that binds there. And then you have another region that would be the DNA binding domain. And then if you have your strand of DNA, so it comes down here and then it binds to the DNA. So this region is where you would find or could find zinc fingers. And then as you can see, you're having this zinc finger in this region of the receptor enhances its DNA binding function. So leucine zippers is another example, and it's formed by leucine residues, as you see here in the red spheres, of one alpha helix interdigitating with the leucine residues of another alpha helix. So you have two here and here, and these interdigitate, and they form what's called a dimer. And you can see that here. So you have one alpha helix here, one alpha helix here, and they're combining together. There's a magnesium here that's coordinating them together. And these are also found as components of DNA binding proteins, such as transcription factors. And again, remember, so you're in the nucleus here, you have your DNA, and you could have RNA polymerase, but then remember there's also transcription factors that bind to the DNA and help enhance this binding, or they can also block it as well. Either way, the transcription factor has to bind to the DNA, and some of them can have leucine zippers, which help enhance that function. All right, so we're going to take a minute here and talk about prions because this is a high-yield topic for biochemistry exams, and they also have a lot of clinical relevance as well, so that makes them a high-yield topic for board exams as well. So what are prions? They are misfolded proteins that become quote-unquote infectious molecules, so they're not bacteria or viruses, but they're infectious in the sense that within the nervous system, when they come in contact with normal proteins, they induce changes in the secondary structure of these proteins that come in contact. And then that converts that normal protein into a prion. And then these prions combine to form filamentous aggregates that are resistant to degradation and heat. So we'll blow this figure up and take a look at it. This explains the mechanism of generating prions. So you have the normal protein, which is named PRPC. So this is a normal protein that's found in humans. So this is found in all of us. It's a membrane protein. And the function is not really fully understood. It's an area of research, so it's still being investigated. There's some theories, but nothing concrete yet. So for your case, you just want to know that it's a normal protein. It's a membrane protein. It's part of normal function that's found in the cell. Problem is it can be converted into prions. And prions are these misfolded proteins, which are called in this diagram PRPSC. So you have the basic mechanism here is you have PRPC, which is normal, which is then converted to PRPSC, which is prion, okay, which is abnormal. So let's go look at this diagram because this explains how this occurs. So you can have three different mechanisms of acquiring these prions. So you can have spontaneous generation of them. So they just spontaneously happen. You can also inherit a mutation in this protein that causes a conversion to a mutant form, which is then a prion. You can also have inoculation, which is where one disease that prions lead to is famously known as mad cow disease from eating prion-infested beef. That's where you would consume meat that would have prions in it already. So you ingest these prions, and then they can come in and lead to development of prion disease in the individual. So regardless of how it gets there, you have this prion, PRPSC. So you have PRPSC here in red, and then normal PRPC here. So it interacts with a PRPC, and then when it interacts with the normal PRPC, it induces changes in its secondary structure of that protein, and that's what converts it into a PRPSC. So now you have two of these. And this is a cycle that keeps going on. So then it's going to keep going, to this one and eventually this one. And, this one gets converted and this one can go to that one. And then this one goes to that one. And you can see where it's, a ongoing process. And so this is a cyclical process that occurs over time. And then you end up accumulating these things. And that's where you get these aggregates. And again, these aggregates, are resistant to heat, and then degradation. And what we mean by degradation is that you have proteins in the cell called proteases, which break proteins down. So it's resistant to degradation by proteases. The other thing to note is that since these are endogenous proteins, the immune system doesn't recognize this. So it's a normal protein, the immune system doesn't recognize this and doesn't mount a immune response, because potentially if the immune system would react to this, it could stop it. But unfortunately, the immune system doesn't recognize it as a foreign invader. That's something that needs to be destroyed. So then where it comes in clinically is that prions can cause fatal neurodegenerative diseases. So when these prions aggregate and accumulate in nervous tissue in the brain, that's what can lead to neurodegeneration. And then these are all fatal. They eventually result in death. So Creutzfeldt-Jakob disease, which is also mad cow, has early symptoms and late symptoms. So early symptoms, you're going to have impaired memory, difficulty with vision. They could develop personality changes and then also exhibit hallucinations. Then it's going to progress over time. Unfortunately, these are not diagnosed during an early phase. Usually these symptoms, especially if it's an older person, someone may think it's, early onset dementia or maybe some Alzheimer's symptoms coming in. But then it develops in the late symptoms where, it continues to cause damage to the brain and you develop more serious neurological symptoms like myoclonus, ataxia, speech impairment, and then they end up developing coma and then eventually they pass away. And so unfortunately, this is a uniformly fatal disease. Then there's another form of prion disease called Kuru. Now this was initially discovered in tribal populations in New Guinea, which is an island near Australia, and these tribes had a ritual where they would participate in human cannibalism and would eat the brains of the deceased. And the thought is that they were contained these prion diseases, and so they would just kind of like ingesting in mad cow disease, ingesting beef with prions. In this case, they were ingesting human brains that had prion disease in them. Now, this is a little bit different. They're asymptomatic for about 10 years, which would be the incubation period, which is where that cyclical process that we described on the previous slide is occurring. You keep accumulating and accumulating prions. So then it develops clinical symptoms eventually, where you're going to have a first stage where you have ataxia, impaired fine motor control, tremors, and dysarthria. Then it develops into the second stage where things start to get worse. You have an inability to walk without assistance. These tremors are getting worse. Then they develop emotional instability and spontaneous laughter. And then eventually it progresses to the third stage, the terminal stage, which is where all these previous symptoms continue to progress and get worse. And then eventually the patient develops dysphagia, they have trouble swallowing, and then chronic wounds that become infected, and then eventually they pass away. So after secondary structure, there's tertiary structure, which is then where we're getting into the three-dimensional folding of the polypeptide. And this is actually considered the native conformation. So you can see this here where you have multiple different secondary structures and they're folding to become, this 3D structure that would be the native conformation. And one thing to note is that the 3D structure of a protein has different sections, which are called domains, that are responsible for specific functions. So we've talked about the steroid receptors. So steroid receptors, they are in the cell, in the cytosol. They bind a steroid molecule, and then they translocate to the nucleus and bind the DNA and act as transcription factors. So they're going to have a ligand binding domain. This is where the steroid molecule will come in and bind the receptor. And then they're going to have a separate domain that'll be the DNA binding domain. So DNA binding domain. And then this is what will eventually bind to the nucleus and help carry out transcription of certain gene targets. So you can see it's one protein, one structure, but it has these multiple domains with different roles to help carry out the overall function. Another thing is the core structure has both hydrophobic side chains, which point toward the center. So they're going to be pointing towards the center, and that makes sense. These are going to be immersed in the cytosol or the aqueous extracellular environment, and so they're going to move out of the way away from water. And then you have the hydrophilic side chains, which are going to point outward into the aqueous environment. So most proteins form tertiary structure and then they're done, that's it. But however, there's some proteins that then also combine with other proteins to form a larger protein that is quaternary structure. So quaternary structure is multiple polypeptide chain structures, so multiple tertiary structures, as you can see here. So here's a tertiary structure here, same thing here. So there's three of them coming together to form one protein, or quaternary structure. And so they form a functional multimeric protein. So this protein together, it needs all these subunits to carry out its overall function. Now there's dimers that contain two subunits, or two polypeptides. And common examples of that are DNA binding proteins. Remember when we talked about leucine zippers? Those were dimers, and they were DNA binding proteins. Tetramers contain four subunits, or four polypeptides. These are actually the most common form, and the major example for that is hemoglobin, the oxygen-carrying protein in the blood. Protein denaturation occurs when the protein loses its native conformation, and this is a result of an external insult. And since you've affected the native conformation, or the structure of the protein, you've impaired the protein's function, because the function is dependent on the structure. And these external insults, they can affect the quaternary structure, they can affect tertiary structure, they can affect the secondary structure, but they do not affect the original polypeptide sequence, or the primary structure. Denaturing agents include high temperatures. That's part of the reason why really high fevers can be devastating, is because they, high temperatures cause protein denaturation, and our body is essentially composed of proteins, and many enzymes and other functional proteins. Extreme changes in pH, or in ionic strength. So these affect the actual environment that proteins are immersed in. And so if an environment becomes very acidic or very basic, it's going to affect the chemical nature of a protein, thus affecting its structure, which would affect its function and denature it. Also, heavy metals such as arsenic, mercury, and lead can also lead to protein denaturation. And that's part of the reason why these metals are so poisonous if they're ingested, is because they can lead to widespread protein denaturation. And again, our body structure and function depends on proteins. So that's why these can be so devastating. Denatured polypeptide chains, so if you have these like this, they can aggregate together to form, like these aggregates or balls of aggregated polypeptides that are denatured, and they become insoluble due to the interactions of exposed hydrophobic side chains. So remember, previously we were talking about how the side chains in a normal protein structure, they're all pointed outward away from the water. In these aggregates, since they're so disorganized, the hydrophobic side chains are not all pointing towards the interior. They're actually, some can be pointed outward like this, and that's what's results in, because you have hydrophobic, it's not going to mix well with water, so it's going to become insoluble. So protein function, as we've said many times in this lecture, is correlated with protein structure, and so therefore, again, altering the protein structure can affect the function. Proteins are also a major secondary source of metabolic energy. So we ingest proteins in our diet, we break them down to amino acids, which then undergo further metabolism, which helps produce metabolic energy. And then proteins can also serve many other critical functions. There's structural elements such as collagen, which help form the ultrastructure of tissues and organs. Then you have membrane proteins. So you have your bilayer like this. You can have a protein that extends across the membrane and help transport molecules into the cell or transport molecules out of the cell. You can have receptors that are embedded in the membrane and bind signaling molecules and transduce a signal. Lots of different functions for membrane proteins. And then you can have carrier proteins in the blood. So if you have a blood vessel like this, and then you have a protein like albumin here, and it can carry, help carry molecules through the blood. Enzymes are a special type of protein, and these are at the center of cellular and metabolic function. So enzymes are essentially biological catalysts for biochemical reactions occurring in cells. And so if you remember from your chemistry days, if you have energy here, and then you have reaction here, so you have your reactants here, and then you have your final products here. So without the enzyme, this is the energy of the reaction. This top point would be the energy of activation. So what an enzyme does is it lowers that energy of activation. And so it helps reach equilibrium faster, but just like any other catalyst, it does not change the concentration of substrates and products at equilibrium. And the thing is a lot of these reactions, they would be so slow, or they almost would just never happen in a cell if it weren't for enzymes, which is why enzymes are so critical. So structurally, enzymes contain an area that is called the active site, which actually binds the substrate. So if we draw a little cartoon here, this would be your active site in here. And if you have your substrate here, substrate can come in, bind to the active site. And when the substrate binds to the active site, it induces a conformational change of the enzyme. So it changes the structure in a sense, because here it's inactive. But when it binds the substrate, it becomes active. So then let's say, just for cartoon's sake, it changes from a rounded structure to a square-like structure when it's bound to its substrate like this. So now it's active. So that's the key concept you want to take away, is that it has an active site, it binds the substrate, induces a conformational change, activates it. Enzyme activation and function are sensitive to temperature, pH, and ionic strength, just like any other protein like we were discussing. So if you raise the temperature, you could denature an enzyme. If you raise the pH, you can denature an enzyme. Again, this is you're affecting the environment the enzyme is operating in. And so if you affect enzyme function, you can end up affecting metabolic and cellular and physiological function, which can be very devastating. And one of the reasons why the ideal body temperature is 37 degrees Celsius or 98.6 Fahrenheit is because this is the temperature at which enzymes in the body function optimally. So this is the optimal temperature for them to do their work. And that's why, again, a really high fever or being really, really cold, really low hypothermia, among other physiological effects, can also affect enzyme function. And so, again, that's why our bodies work hard to make sure this temperature is maintained. So you can have what's called inhibitors, and they can bind to an enzyme. So if we have an enzyme structure like this, and you have your active site here, and you have a substrate here. So you can have an inhibitor I. Inhibitors can actually bind to the active site, and so they block the substrate from getting in. That's what's called competitive inhibition, and we'll talk more about that in the enzyme kinematics lecture. You can also have what's called allosteric, either inhibition or activation. So if you have an enzyme structure like this, so you have an allosteric site right here, we'll call it allosteric so you don't confuse that with the active site here. So you have an inhibitor that comes in, and then you have substrate. So the substrate can come in here and bind, but the problem is if you have an allosteric inhibitor, it can come in and bind and change the conformation to where, you know, it's like so. Again, changes the conformation and makes it harder for the substrate to bind and harder for it to become active. It could also just render the shape of the protein just in a conformation that's not ideal for overall function. It may not necessarily even affect substrate binding. Now you can also have allosteric activators, and these are molecules that, again, if you have our enzyme structure like this, so you have your allosteric site here. Let's say you have an activator A, that comes in and binds, and that puts it into an active conformation, where then the substrate can come in and bind and it becomes active. And again, the thing with allosterism is it's not binding at the active site. So this is an area outside the active site, and it enhances the enzyme function. And it can enhance it in many different ways. It's not necessarily enhancing substrate binding. That's one way it could do it. It could just be enhancing the enzyme function just by putting it in the most ideal conformation. The other thing are coenzymes. Now these are non-protein molecules that bind to enzymes, and they enhance their function during certain reactions. So a lot of enzymes in metabolism especially are reliant on vitamins, and that's part of the reason why vitamins are such an important part of our diet. NAD+, and then tetrahydrofolate. So these are molecules that can bind to enzymes and enhance their function as well. And in fact, there's several enzymes where they really won't function unless they bind one of these, because they need to be just put in that peak conformation. All right, so that wraps up our discussion of protein structure and protein function. In the next lecture, we'll talk about translation and protein synthesis.